Hello, everyone. My name is Chris Bathus. I'm the Chief Commercial Officer at AnaBios. It's my pleasure to be the moderator for today's webinar. People are still logging in, so we'll give them a moment. In the meantime, you can look at the handout section in your panel and notice there's different brochures and publication that our scientists did in collaboration with Dr. Urban and his colleagues at Novartis that you can download and start taking a look at. Today's webinar will have two speakers, Dr. Urban and my colleague from AnaBios, Dr. Najah Abi-Gerges. And the title of today's webinar is Preclinical Assessment of Native Human Cardiomyocytes Provides Translational Value in Drug Development. I'm just going to say briefly a little bit about AnaBios, and then Najah will take a deeper dive in his introduction. Those of you that don't know us, we are a preclinical CRO based in San Diego, California. We recover human tissue samples from ethically consented donors and use the tissue or cells to perform physiological assays in which we can test preclinical compounds for drug discovery projects. So what we've essentially done is redefined first in human. And as far as doing preclinical research, it's really as close as you can get to clinical trials without actually doing a clinical trial. Now, before I introduce Najah, I just want to take this opportunity to say a few words about a translational research grant we're doing for the second year in a row. Those of you in academic labs, and I know that many of the audience members are from academic groups, even those from biotechs and pharma collaborate, so make sure your colleagues know about this. We are offering a grant of up to $15,000 in high quality human tissue samples for academic research. And the application is quite easy. You just submit an abstract to us. The deadline is June 30th, so go to the AnaBios website for more information. Okay, so now let's get started. It's my pleasure to introduce Dr. Najah Abi-Gerges. He's been with AnaBios for 7 years. He's the Vice President of Research and Development. He holds a Master's of Physiology and Pharmacology from the University of Poitiers in France and a PhD in Cardiac Physiology from Paris University in France. Prior to joining AnaBios, he was involved in drug discovery programs at AstraZeneca, and he has over 17 years in the pharmaceutical industry. He's an innovative leader, having made substantial contributions to drug approvals. He's published over 40 peer-reviewed articles and contributed novel paradigms to advance cardiovascular translational science, including AnaBios' unique human primary cardiomyocyte platform. He's also the editor for the Journal of Pharmacology and Toxicological Methods, and has been a reviewer for more than 20 peer-reviewed scientific journals. He's also the co-chair of the Ion Channel Group in the CEPA initiative, which you'll hear some about today. So, Najah, the floor is yours. Take it away.
Yeah, thank you, Chris, and welcome everyone to today's webinar. As Chris said, at AnaBios, we conduct human translational research, and the models we have been developing at AnaBios allow drug discovery teams to study the effect of novel drugs in human cells and tissue. Of course, when it comes to drug-induced toxicity, AnaBios models provide critical data that allow teams to determine if novel drugs are safe in a human. Secondly, if novel drugs, or to help define a safety margin for this novel drugs. And thirdly, the models help to select those for first in human studies. To have access to ethically consented organ for research, AnaBios works with organ procurement organization in the United States. As shown on this slide, the critical element of our procurement success can be summarized or based on standardization of method, prevention of tissue damage by avoiding ischemia, and of course avoiding reperfusion injury. We have access to large network that ensures a high volume of organs. And finally, we have extensive annotation for each organ. One of the organs we recover is the entire human heart. Next slide presents AnaBios' current cardiotoxicity assessment offering using normal donor hearts. As you can see on these slides, we run tissue-based models to support projects during late preclinical development, and also we run cell-based models to support optimization of drug during early discovery. The data generated with these models help discovery teams to assess the potential of novel compound to affect the ECG, to affect the myocardial contraction, to modulate heart rate and blood pressure. For today's webinar, I will focus on the cardiomyocyte contractility model, so my talk can complement Dr. Urban's presentation. As you see on this slide, we have developed at AnaBios an isolation protocol that ensures that we get a high yield of functional cardiomyocyte. You can see on this slide an image of representative typical adult primary cardiomyocyte. This cardiomyocyte, they display the rod-shaped morphology with a clear striation. And most importantly, this cardiomyocyte do not contract spontaneously. They require application of an electrical field stimulation to contract and relax. So basically, to influence optimization of drugs and help exploring structure-activity relationship, we believe a contractility system with the following characteristic is really required. As you can see on this slide, the system, we believe, should be easy to use. The system should allow recording from multiple myocytes in parallel, so a semi-throughput drug testing can be performed. The system should also allow simultaneous assessment of cell health, physiology, and pharmacology. We want the system when it comes to data analysis, so the analysis can be automated and can be rapid. And also a drug concentration effect of a curve of a drug should be completed within 30 minutes. And finally, the system should be associated with good reproducibility when it comes to data. So we are pleased to announce that AnaBios has developed this system, and we named it the MyoBlazer. This slide presents a video that shows contractility recording from multiple cardiomyocytes using the MyoBlazer. So I will play the video for you. As you can see here, we are recording contractility from 9 cells, from 9 cardiomyocytes in parallel under baseline condition. And here the cells are being stimulated at the 1 Hz pacing frequency. Next, you can see the decrease in contractility that was caused with the application of 10 micromole of verapamil. Verapamil, as a reminder, is an inhibitor of calcium channels. Also, we just finished the development of analytical tools that allows to analyze one run of multiple cells in a few seconds. The tool analyzes different parameters, as you can see on this slide, that can be derived from the contractility transient. Such parameters represent the systolic and diastolic phases of the cardiomyocyte contraction. The cardiomyocyte contractility model has been well validated, and our validation studies have been published in peer-reviewed journals, for example, in Frontiers in Physiology, in Scientific Report in Nature, and in Tox Science. In the next few slides, I will present new data that reconfirms the high quality of the cardiomyocyte contractility data that can be generated with the MyoBlazer. As you can see on this slide, we show data we obtained with the isoproterenol, a non-selective beta-adrenergic agonist. Data obtained from 16 different donors at a concentration of 30 nanomole of isoproterenol demonstrates that iso-induced increase in contractility is largely independent of isolation and donors. Like iso, the interdonor variability for the increase in contractility with the application of 3 millimolar extracellular calcium chloride in 10 different donor hearts was also found to be low and to be independent of isolations. Here is another set of myoblazer contractility data that was obtained with dofetilide. As you know, dofetilide is a hERG channel blocker with application of extracellular calcium with ISO and also with omecamtiv mecarbil, an activator of the myosin ATPase 2. Please note that this compound is now in a clinical development for the treatment of heart failure with reduced ejection fraction. Also, we are showing data with mavacamten, an inhibitor of the myosin ATPase. This compound has been recently approved by the FDA for the treatment of obstructive cardiomyopathy. And finally, we are showing data with verapamil. Again, the data on with this many reference drugs demonstrate low interdonor variability, and the effects of these drugs on contractility were found again to be largely independent of isolation. As indicated earlier, the myoblazer allows to derive kinetics data from the contractility transient. As you can see on this slide, the treatment with omecamtiv can modulate the kinetics of contractility, like rise time and time to 90% relaxation. And such modulation, again, was seen in cells from 4 different donor hearts. However, application of extracellular calcium chloride was found not to affect the kinetics of the contractility transient. Taking together, the omecamtiv and extracellular calcium data, it can be suggested that modulation of the kinetics can be specific to some drugs, but not to all treatments. Data in this slide shows again low variability in mavacamten concentration effect curves, and therefore reconfirm again the low interdonor variability of cardiomyocyte contractility data. Intradonor variability was also found to be low, and here in this slide we are showing data generated with BTS, another inhibitor of the myosin ATPase. The data to be shown in the next 2 slides will demonstrate how human cardiomyocyte and the myoblazer can be used to explore structure-activity relationship. To this purpose, we tested blebistatin and 3 of its derivatives. Note that blebistatin is another inhibitor of the myosin ATPase. As you see on this slide, blebistatin and hydroxyblebistatin share similar ability to reduce cardiomyocyte contractility. However, para-aminoblebistatin and aminoblebistatin show different contractility profiles compared to blebistatin. In summary, data generated with blebistatin and its analogs suggests that the human cardiomyocyte contractility SAR data have the potential to impact chemistry design during early discovery. Please note that all the new data we presented today are to be submitted soon to a peer-reviewed journal for publication. To conclude my part of today's webinar, AnaBios data confirms the ability of the human cardiomyocyte contractility model to detect the potential of drugs to cause cardiotoxicity. We believe the model can be employed during early discovery and during late preclinical development. It can also be used to generate follow-up data to support the clinical programs. One strategy can be to employ the human cardiomyocyte contractility model in conjunction with traditional S7D models. We believe such combination can provide pharmaceutical companies with a unique translational opportunity to rank novel drugs from most to least cardiotoxic and to well-define a human safety margin. Dr. Laszlo Urban will now present Novartis' experience in using the AnaBios human contractility model to support the company discovery programs. And thank you.
Thank you very much, Najah. That was excellent introduction. We will hold the questions until the end of the webinar. Just a reminder, you can input questions in the questions tab in your GoToWebinar panel. And also just a reminder, you can download AnaBios brochures and a paper that was published last year in collaboration with Dr. Urban's group. So what I'd like to do now is introduce the next speaker, Dr. Laszlo Urban. I've had the pleasure of knowing and working with Dr. Urban for the past 20 years, where he's been at Novartis in Boston. In fact, Laszlo has been one of the great constants in my life. I've been at different companies, but I've really enjoyed working with him. I find him to be a real pioneer in using new technologies. When I was at Sophia Bioscience, he brought in one of the early versions of the Q patch and very similar strategy with the cardiomyocytes from AnaBios in the past 5 years. So Laszlo is the global head of predictive safety pharmacology at the Novartis Institute for Biomedical Research, or NIBR, in Cambridge. He's been with Sandoz Novartis since 1990, leading various global pharmaceutical research areas. As the founder of the predictive safety profiling at Novartis, he developed and implemented the early in vitro preclinical safety profiling program for NIBR. He and his team are developing safety assessment tools for decision support, which provide access to integrated information for adverse drug reaction mitigation. Laszlo received his MD and PhD in neuropharmacology from Debrecen University in Hungary. He is honorary lecturer of the University College London and was a visiting professor at Duke University. And the title of his presentation today is Preclinical Assessment of Native Human Cardiomyocytes Provides Translational Value in Drug Development. Laszlo, take it away.
Thanks very much, Chris, and thanks very much for the opportunity to present these applications today from AnaBios. I hope that you can see the figures now. So with that,
I can see it.
Thanks very much. So with that, I dive into the agenda and hope the slides will move. Yeah.
Laszlo, the audio is a little off. Maybe you adjust your microphone.
Is it better now?
Yes, thank you.
Okay, thanks very much. So taking the agenda, I would like to talk today about 3 topics here. The outline of the cardiac safety testing in the preclinical space for the pharmaceutical industry, very briefly, because most of you are familiar with this, or all of you. Then the application of the CardioPRIME assay in drug discovery, how at NIBR, Novartis, we are using it, and give a bit of flavor of the evaluation of proarrhythmic effects of hydroxychloroquine concerning the application in COVID-19 patients. The guidance for testing proarrhythmic events in the preclinical space by ACHS-7B is well known, I think, to all of the audience. I just wanted to highlight 2 parts of it. The one, which is the in silico computer modeling, which has been developed during the past 10 or 15 years, and now it's broadly applied and basically enables us to combine all the ion channel effects in a single artificial cardiomyocyte and see whether they would provide any proarrhythmic effect on the compounds which we are testing. But these assays still don't give the ideal composition of kinetics, and many of the applications don't have the PK part. So it is definitely very useful in terms of clinical predictions in the preclinical space, but what has been developed during the same period of time brought us much closer to the high predictive value, were the human stem cell derived cardiac myocytes, which are fairly broadly used. But today, I would like to highlight basically how do we pull all this together and then top it up with the adult human cardiomyocyte assay. So this is the flow chart, not all the details, but the major points, what we are using in cardiac safety profiling, in the cardiac safety profiling portfolio at Novartis. So I put it into 3 segments here. The very early one, where the step 1, the entry point, is the scaffold selection in drug discovery. And most of the companies, or all of the companies, are using QSCR or associated in silico elements. We use Cube and the HER-Q patch for the analysis of HER, sodium, and calcium channels. in different kind of composition because the frequency of these effects are very different. hERG is a very high, hERG inhibition is a very high frequency event, while sodium and calcium is much, much less. We then feed the data into the in silico model, also apply the pharmacokinetic information which we gain from in vivo studies, and try to predict basically what kind of output we have, whether there is any QTc potential of the molecules. From here, we move to the more analytical phase, which is the middle piece here, and that's where I would like to focus today. So the step 2, the entry point, the lead optimization and candidate selection. For a high throughput or higher throughput applications, we use the iPS cell-derived cardiomyocytes, the calcium transient duration assay in a 384 format, and the MEA assay in the 96 format. Both of them obviously are using the young and, so to say, to some extent, immature cardiomyocytes. The reason we basically moved away, not moved away, but additionally added the adult cardiomyocytes is a very simple perspective. These cells or cell cultures improved during the past couple of years enormously, but still there is a certain immaturity, and we have to count with that. They are predictive in many ways for predicting cardio safety effects in the clinic. But there is one important element, which is that the hERG channel contributes to the maintenance of the baseline resting potential. So if we have a strong inhibitor, this certainly will be offset, and that would bring in unwanted effects. Most of the people believe, who are not in the cardio safety or cardiology space, that cardiomyocytes contract by themselves, which is not true for adult cardiomyocytes, but the iPS cell-derived cardiomyocytes do so. The other element here is what I'm going to talk about today, is the application of the in vitro adult cardiomyocyte contraction model, the AnaBios model, with the adult cardiomyocytes, and we measure inotropy, which in our belief it's really the best biomarker for proarrhythmia and inotropic effects. And we can measure also, obviously, the calcium transients, which go beyond the effect on the particular ion channels. And this system, I hope I will convince you, is really predicting proarrhythmogenic effects. And the last bit here is basically just the regulatory requirements, the GFP hERG, the non-rodent telemetry. So I think Najah was talking about the system already, the CardioPRIME. I just would like to highlight again that here we have clear biomarkers for the proarrhythmic risk. There you can see on this particular segment that dofetilide at the lower and the higher concentration causes after contractions, and then contraction failure, which would be equivalent to Torsades. And also one can clearly measure the contraction in terms of the inotropic effects. So we believe that the advantages certainly are the mature cellular assembly of electromechanical coupling. So there is no spontaneous beating according to that. Both ventricular and the atrial cells are available from AnaBios, and we are using both of these. And this is applicable for compound prioritization, which I would like to show in the first part of my presentation. And these are validated for clinical translation. Again, we will have some examples for that. And finally, it's not an invasive technology. So this is a complex slide, and I partially covered some of the details. So whenever we are dealing, or not dealing, but associated with any other company which provides absolute important information for us, we do a validation study. So this validation study was very similar to what SIPA did in different type of assays with the AnaBios assay. What you see here is basically a table where we had full information on cardiac ion channels, the hERG channel, the sodium, the calcium channel in individually expressed cell lines. And then on the other side of this table, you can see that we also listed the label, the confirmation, in vitro or in vivo for these particular molecules, drugs or preclinical compounds, and the mode of action. So it's basically a very diverse set of molecules which we applied here, and they were all blinded when we started this study. So AnaBios provided the middle part of the data, what you can see here. We also list here the safety margin, which is the IC50 of the leading or the most prominent ion channel effect over the free plasma concentration, which we use Cmax continuously in these studies. And these are clinical data, by the way. So the first segment was basically around those molecules which have high effect, or very potent effect at the hERG channel. Then the second and the third were basically mixed bags with different kind of activities like calcium channel blockers, if you look at the right-hand side. And then the last bit was basically a list of compounds where we had either sodium channel blockers or different kind of different kind of targets which contribute to cardiac contraction or proarrhythmogenic effects they have. From this study, we basically got the information that if we apply two concentrations, and the reasons we use these because we wanted to use it at a very early phase of drug discovery, so it should fit into the lead optimization phase. That two concentrations, the 0.3 micromole and the 10 micromole, gives us two perspectives. The 0.3 is very close to most of the drugs, what they have as a therapeutic free plasma concentration, and you will see it in a later example in the clinic. The 10 micromole was the reason that we wanted to know what is the therapeutic window, or in the preclinical space, we call it basically the safety index or safety window. So what you see here is basically either at 0.3 micromole or at 10 micromole, we see proarrhythmogenic effects of all the compounds where it was expected. And we saw also some negative effects at 10 micromole, which is way above the exposure concentration, usually in the human, in patients. You can see also that the safety margins here, which would be expected if the compound is safe or the drug is safe over 30, that is way beyond, with one exception, under 30. The other groups performed pretty well. You can see that again, where we were expecting negative data, they came through with one exception here, phenytoin, which is quite interesting data that at lower concentration, there were some proarrhythmic effects, but at a higher concentration, they disappeared. And with fexofenadine, we saw it at higher concentration, but not in the therapeutic range. And as you can see, the safety margins here are pretty large. So there is no expected effect in the clinic. If you look at labels, there are no labels here. But in some cases, for example, with ranolazine, there is an QT prolongation warning, even though we don't see any effect in the preclinical space. And that was confirmed also by other studies. The last segment here are proprietary compounds to Novartis, and I highlighted on the right-hand side that they came from different projects, GERK inhibitors, kinase inhibitors, and TRPC6 inhibitors. Certainly here we don't have data from the clinic, but what we basically confirmed that our GERK inhibitor was absolutely negative in the ion channel assays, and it was also without any effect in the AnaBios studies, and we didn't see any effect in our experimental work either, and in late preclinical in vivo experiments. However, with the kinases, we have a mixed bag of ion channel activities. We see QT and QRS changes, and these were also confirmed here in the AnaBios studies. And notoriously with the TRPC6, we had issues when we went to in vivo experiments and saw some arrhythmia at higher concentrations, which were also basically confirmed by the AnaBios assay. So taking all this together, I took a bit of time on that, all these thorough analysis provided us all the confidence that we would be able to use this assay basically for two reasons: for translating the data from preclinical to clinical, but also with these two concentrations, we can go in at earlier phase during lead optimization and can do rank ordering, which is absolutely essential for the development, for this development phase. So I would like to show you a segment from one project, which is a quite interesting one, that here we basically rank ordered molecules, those molecules or compounds which did not have at all any effect at the 3 ion channels we examined, hERG, sodium, NaV 1.5, and CaV 1.2, but we saw some cardiac effects. So what you see here is that basically we recorded at 3 and 30 micromole concentrations. These were related to the higher therapeutic, multiple therapeutic dose equivalents. And we basically got the inotropic data and also the arrhythmogenic effects or the chronotropic data. So what you see here with compound A, there is a time-dependent contraction increase. And at 30 micromolar, it doesn't increase much further, so it reached peak at 3 micromolar. These are the times 10, 20, and 30 minutes in comparison to vehicle. And as you can see, at a higher concentration, at 30 micromole, we have very clear signs of proarrhythmic effects, like after contractions, and contraction failure also developed at 20 minutes application. So we saw these proarrhythmic effects, and these were confirmed in vivo QTC experiments. So we deprioritized this molecule. Compound B is somewhat different. Here we see much less effect on inotropy at 3 micromolar. At higher concentration, we do see some increased effect, but the concentration dependence is certainly questionable. But what you see here is very importantly that this molecule caused after contractions to great extent at both concentrations. And again, we had QTC data and we deprioritized this molecule. And finally, I show you the compound C, which has inotropic effects at 3 and positive inotropic effects at 3 and 30 micromole, but there was no sign of any arrhythmogenesis. And in this case, this compound moved further on, and we have confirmed clinical cardiac safety margin. So that concludes basically the experiments which we did for rank ordering. We are using this assay now for a particular type of molecules where the cardiac side effects are not necessarily associated with ion channel direct effects, but probably effect on the calcium homeostasis or some other elements of cardiomyocyte inotropic and chronotropic functions. With that, I switched to a more clinically related study, which we did with AnaBios. The Lions part was done at AnaBios, all the experimental work supervised by Najah. And this is about the cardiotoxic potential of hydroxychloroquine, chloroquine, azithromycin in adult human primary cardiomyocytes. And you know the hype behind it during the COVID pandemic, the existing COVID pandemic, and the debate whether this medication was, number one, potent or efficacious and safe. So what we wanted to know to determine the concentration dependence of these effects, establish the human safety margins in vitro, and assess the influence of risk factors. We knew that the ion milieu of the patients change, and temperature changes are also expected. There is an inflammatory condition, and whether there is magnesium concentration elevation is used to amend some of the effects. And because HCQ was hugely used together with azithromycin, the combination was also basically looked upon. We had the ion channel effects of these 3 molecules, as you can see here, that azithromycin was not much effective up to 300 micromole in hERG and now 1.5, but it had a weak effect at the calcium CaV 1.2 channel in these assays. Chloroquine and hydroxychloroquine, on the other hand, were effective at hERG, and chloroquine in all the 3 assays, while hydroxychloroquine had an effect in the calcium assay. So here are data, and you can consult these in details because the papers are also provided, or the paper was also provided. So you can look into the details. I would like to take you through pretty quickly on the upper line. So here we have basically HCQ, CQ, and the combination of HCQ with low potassium with 2.5 micromole. What you see here in consequent figures is the green shadow, which shows the human therapeutic effective plasma concentration. And this is a pre-plasma concentration. So we try to basically position our experiments so that we cover this area and then we go above, so we can give basically a margin for safety. And that's what you see on the columns here. These are, the first line is the concentrations, the second line is basically showing what is the margin. So obviously the green area is the 1 times, and then we go up to 30 times. And what time was the measurement done? You can see that HCQ, if you look at the therapeutic range, it doesn't have tremendous effect, but it has triggered contractions at higher concentrations. The after contractions appear. Different picture we have for chloroquine, where we see strong after contraction. It's over 10%, but with a large error bar. And if we increase the concentration, then certainly the other biomarkers for arrhythmogenesis appear. And if you compare C to A, then you can see that the appearance of after contractions is pretty frequent when we do the combination of low potassium concentration with HCQ. In the lower part, we applied high magnesium in addition to HCQ, and you can compare it to the one above and the one on the upper right-hand side, that we don't see in the 1 times margin any kind of sign for arrhythmia. And if we go up to 10, then very little up or below 5% we can see. Azithromycin was tested on its own first. Again, you can see that there are after contractions associated with azithromycin. And if we do the combination, which is pretty much relevant for what the clinicians are doing, then we can see that there is an increased appearance of after contractions mostly, and contraction failure at much higher range in this particular combination, highlighting that in the clinical application where HCQ and azithromycin was used, the potential risk is increased. So this is just a snapshot of some of the raw data. The upper and the lower segments basically correlate with each other. And you can see here 3 different type of proarrhythmic events, or 2 after contraction and the premature contractions. And with HCQ, the effect is much less, but you see at 10 times the therapeutic free plasma concentration, the regular appearance of after contractions, so representing basically after depolarization. With CQ, that's much more prominent, and certainly the premature contractions come here, and this is only 3 times in comparison to the 10 times of hydroxychloroquine. And finally, you see erythromycin, where you can see also some of the after contractions, but less pronounced if it's on its own. We also wanted to know about the effect of temperature. And basically, these experiments are done at 35 centigrade, and we were able to go up to 39 centigrade. And you can see that in terms of the incidence of after contractions increase significantly. But if we combine HCQ with temperature, in comparison of the 2, there are no significant changes. So this is not a major player in the enhancement of risk of arrhythmia. I would like to show you this figure because in a snapshot, this gives you a pretty good visualization tool on the summary of these findings. So these are the different kind of biomarkers: the after contraction, the contraction failure, triggered contraction, and post-dependent arrhythmia. And you can basically immediately realize that the after contractions are probably the best biomarkers for this particular experimental work. And here it's very clear that if we look at the therapeutic index, then 1-3 times azithromycin, chloroquine, and the combination of hydroxychloroquine and azithromycin could cause an increased risk for arrhythmias. And this is particularly true for HCQ. And considering that many times there is polypragmasia, so the application of many other drugs which might have effect, for example, on CYP3A4, could make and enhance the appearance of both of these drugs in the circulation. If you look at the other biomarkers, there are not these prominent changes. So we were guided by these after contraction % incidence. And these are just the more thorough analysis of the data. Now going into the IC50 values. So this figure A is basically giving you the IC50 values of the different molecules we tried here on their own. And you can see, again, we marked the therapeutic concentration range here. Then if we are going now to add to the HCQ curve, which is equivalent to what you see in A, azithromycin, then you don't see any effect in terms of the inotropic effect. So the inotropic effect is basically no minimal or no decrease or increase. Then we did a combination of HCQ with azithromycin in 2 ways. So we basically, what you see with the green dots and the line, that here we had the different concentration of the drug in the presence of 0.3 micromolar azithromycin. And interestingly, you can see that basically the curve straightens out under these conditions. So there is no change, no decrease in inotropy. If we do it the other way around, so we basically dose azithromycin and then add a stable concentration of 0.3 micromole HCQ, then there is an increase here in terms of inotropy. The lower segment is basically echoing what you see above, but here we don't use the drug concentration. Here we use the multiple of the therapeutic free plasma concentration, which is the Cmax. And you can see that this correlates very well with the above lines. So this is basically what we published in the paper, and there are many more details which you can consult later. But what I wanted to add to this is, because there's a huge number of publications, pros and cons, but I basically used 2 of the meta-analyses recently appeared. One is, which is basically looking at the various clinical trials and concludes that the benefit using hydroxychloroquine or the combination with azithromycin is much, much less than the increased risk of arrhythmogenesis in COVID-19 patients. The second paper here is very similar, but it's using a different resource, which is the FAERS, the FDA Adverse Event Reporting System. And it comes to basically the same conclusion. And it's added also as they analyzed lots of information provided voluntarily to the system, that if there are any other drugs, like some which could inhibit CYP3A4, could cause more trouble, as the concentrations of hydroxychloroquine and azithromycin might increase under these conditions. And that's what we basically demonstrated in the previous figure. So in terms of the translational value, we were able to demonstrate how under a particular condition, these changes would affect the patient. And this is an interesting aspect because there are other applications of hydroxychloroquine, for example, in rheumatoid arthritis, where on the different indication, there might be a combination of different drugs. it doesn't diminish the usability of hydroxychloroquine in prevention of malaria, where it's used primarily. And on that condition, where most of the treated people are not patients, but healthy people, the risk of arrhythmia is much less. So the conclusions and perspectives we come to. So we demonstrated the mechanistic and translational value of human cardiomyocyte contractility-based model, which is the CardioPRIME as a key component of drug discovery, applicability to that. And in this study, we were the first to show the profile of proarrhythmia risk and inotropic activity of HCQ, CQ, and azithromycin and the combination and the different environmental changes. And the data generated with these three drugs have the potential to inform studies evaluating repurposed therapies, as I just mentioned before, including those of COVID-19. And we are at the moment further exploring the utilities of adult human cardiomyocytes. And these studies are basically using trabeculae and also the atrial cells, because all of these, what I presented, were obviously on ventricular cardiomyocytes. And try to dig a bit deeper with proteomics and RNA-seq in the same experiments at different exposure times. And certainly, we are now ready to explore the application of MyoBlazer platform, because we then have access to achieve increased capability, capacity, and reduce cost. And what leaves me with is basically the acknowledgement to the great contribution from AnaBios. I'm particularly grateful for Najah, the lead scientist on this hydroxychloroquine project, but also Paul, Andre, and Chris contributed significantly to this study, and the neighbor team, Pierre Jordan, who was our clinician leader in the team, Martin Berenger and Anatoly, who were basically partners in crime, Anatoly providing the ion channel data, and Martin and Berenger basically contributing a lot to the development of the paper. With that, thanks very much again, and I'm ready for questions.
Thank you so much, Laszlo, for an excellent presentation. We do have time for questions for Laszlo and or Najah. You can enter your questions in the panel, the GoToMeeting panel on your right, or you can simply raise your hand and I can unmute you. So we'll start with Najah. Najah, how does the MyoBlazer data compare with data from experiments with trabecula, ventricular trabecula?
You mean when it comes to contractility and proarrhythmia data?
Yes.
Yeah, I mean the correlation is good between the two, and you know, there is a difference when it comes to effect on contractility when you have multi-ion channel blockers. You know, there is a little bit of shift in the dose response curve between the two models, but the correlation between the two is pretty good in general.
Okay, thank you. I have a question for Laszlo. You mentioned at the beginning of your presentation the CEPA initiative, which really started in discussion phase in 2013. It never has become an official guidance as the S7B was. It's more of a flow chart or whatnot. Have you seen, I think the original goal of CEPA was to rescue drugs that may have been left out in the past. Have you seen that happen at Novartis during your time there? Has it helped to rescue compounds, or has it provided added value for your team?
It's a very good question, because certainly all the elements we are using here in our assessment, preclinical assessment, I think there is a value that we have more variety of assays, and we can rely on higher confidence composing all of that data. What I showed in our flow chart in the second box, we are basically do the combination of iPS cell-derived cardiomyocyte data together with the in silico, and then basically what findings we have here, we can apply to those molecules which have the potential for further development, and then we can test those molecules now in the human adult cardiomyocytes the way I showed. And this I think this aligns with what the CIPA initiative suggests to do. I think we did the extra step with the adult human cardiomyocytes, but I think it's basically a new paradigm that people are using to much greater extent, the cardiomyocyte assays.
Excellent, thank you. Yeah. So we have more questions coming in. Just a reminder, you can either raise your hand and ask verbally, or you can type in your question. And also during this time, you can download AnaBios brochures and the publication, the HCQ public paper that Laszlo mentioned. Okay, the next question is for Najah. How do you determine the magnitude and modulation of the voltage applied on the cells?
Yeah, basically we look always for a threshold, basically. So you go from a threshold where you don't see you don't induce a contraction, basically, and then you ramp up this threshold until you find it, and then basically you go above this threshold with a certain percentage to maintain the contraction during the entire experiment.
Okay, and here's a question from Igor Efimov. He's got a couple questions. Have you started using human cardiac slices yet?
No, as Igor knows, we are collaborating with Dr. Tamer Mohammed at Lowell University. So we will start hopefully soon with with slices from normal hearts, and then we will move to use slices from diseased hearts.
Okay. And he also wanted to know, what are the plans with recording of action potentials and calcium transients?
In slices or in myocyte?
I guess you can
anyway,
say
for the myocyte, we can record action potential using current clamp, and we can run calcium transient. So we have a similar model to the myoblazer when it comes to the calcium imaging. We can record from many cells in parallel. When it comes to the slices, recording calcium and action potentials, this is something we are still discussing with Tamer, but the priority will be to start with contractility.
Excellent. Just a reminder, if you join the webinar late, it's been recorded and will be available online within a few days. Here's a question. Can you comment briefly on the methods that allow one to measure changes in sarcomere length in fractions of microns? Najah?
Yeah, basically we use bright field imaging here, to record or follow changes in sarcomere lengths. And as I showed briefly, in one of the slides, this contractility transient can be analyzed, and many different parameters can be derived, and these parameters can be used, to differentiate between compounds and to rank compounds too. And it can provide also some mechanistic understanding of the effect of a drug.
Okay. And here's another question for you, Najah. Is there a way to evaluate proarrhythmic risk of compounds that occur only after chronic exposure?
Yeah, I didn't show the data today, but we developed a new protocol that allows us to keep the myocyte in culture for 24 hours, but keeping them for 24 hours, they don't change morphology and they don't start to beat spontaneously. So we use electrical field stimulation, and we can keep them 24 hours. And yeah, we can use them, to assess the effect of compound for 24 hours treatment. And we are doing now two big projects for two big pharma, using this models. But also we have the plan when we start using the cardiac slices, the preliminary data with the normal slices that can be maintained for up to 12 days. So the hope here is that we can look for on the same slice for acute and long-term effect of drugs. And when you keep the slice for many days, you can not only look for acute and long-term effect of drug, you can look at change in gene expression, you can look at biomarkers, proteomics. So there is there is a lot of opportunities that can be done, with a slice model.
Excellent. And the same person asked, which mode of action would be related to drug-induced defective ion channel trafficking? Is that visible in our model?
Yeah, we haven't yet tested, compounds that, for example, will induce the trafficking issue with the hERG channel. But this is something we would like to test, and also start looking at the long-term effect of cancer agent too.
Excellent. We have a question from the audience, Dr. Simon Shea from AFSI. You'll need to unmute, Simon. Looks like I can't unmute you. We'll come back to you, Simon. There you go. Go ahead, Simon.
Yeah, thank you so much. From the NIBR group study, it looked like the whole adult cardiomyocyte will be predict or at least more sensitive and actually to reflect the clinical liability of the potential cardiovascular side effect compared to a single 3 ion channel. The data you've been presenting, hERG sodium channel 1.5 and calcium channel 1.2. That might understand correctly, because if we base it on these 3 typical ion channel, IC50, and compared to the therapeutic doses of free concentration 0.3 micromolar, then you predict, yeah.
So is this a question for Laszlo? You're basically asking if how the cardiomyocyte data compares to the data from the ion channel studies. Is that right?
Yes, that's correctly. Yeah.
Go ahead, Laszlo.
Yes, certainly. There are 2 aspects. Let me go back a little bit what Najah said about the possibility to predict longer lasting effects like trafficking, and what is well known now. We published also in other companies on HDAC effects, where maturation of the hERG channel is affected by HDAC inhibitors. It seems like both the iPS cell-derived cardiomyocytes and the adult human cardiomyocytes are able to predict, and that's an interesting observation, and we are in the middle of finding out why is this correlation so good within a brief period of time. So that's an addition to what Najah already said, that in short-term evaluation of these cells with these particular compounds gives us a pretty good prediction value. Now, answering your question is that yes, if we have a mixed bag of ion channel inhibitions, plus the molecules might also affect calcium homeostasis, the electromechanical coupling, then these cells are the ideal ones because they have the mature system or mechanism for all of that. And that's why we basically choose these 2 concentrations, the 0.3 and the 10 micromolar concentration, for practical reason that we definitely can differentiate between the expected effect at the therapeutic level, and then we can also have an idea of what would be the safety margin even before we have pharmacokinetic data, or we have only preclinical pharmacokinetic data. So how does it compare to the iPS cell-derived cardiomyocytes? We use iPS-derived cardiomyocytes for a much larger scale screening earlier. And then, as I mentioned in my talk, we are fine-tuning for better understanding of inotropic effects, which we haven't seen, for example, for those compounds which I showed you, the 3-1, where I did the rank ordering in the iPS cell-derived systems like MEA and calcium transient assay, but it was clearly visible in the adult human cardiomyocytes.
Great. Thank you so much for your very. May I ask one very simple question? Have you examined any of your drug or the portrait control drug on the SA node? Because that would be truly control the heartbeat. We're not talking about the spontaneous muscle contraction, but we're talking about the the SA node. Yeah.
Yeah, I'm not 100% sure I understood the question.
He's asking about the, have you done any experiments testing your compounds on in SA node assays at AnaBios? I think the answer is no.
Not yet. Yeah, sorry, I got it. Yes.
Okay.
No, we haven't done it. We haven't done it yet. This is something very important question, and we have a certain disease area, cardiovascular, which is very much interested in this, and definitely we are interested also. There were some kind of applications under development a couple of years ago, which addressed this question in a different system, but they failed. So there is a gap, and instead of going into a isolated heart, perhaps this would be a much closer shot to the clinical situation. Thanks very much for this suggestion. It's certainly something we consider.
And speaking of SA node, Yusheng Q from Amgen asked if we use cells or tissues, and is there validation data that we can share? Most certainly. Najah, can you comment on that?
For the SA node data?
Yes.
Yeah, we have data, with a few positive chronotropes and negative chronotropes, including compounds that have been in a clinic. Yeah, we can share this data with the team at Amgen and with Yusheng. But there is, yeah,
with tissue, correct? That's a
yes,
tissue.
This is SA node, the tissue sharp electrode recording of spontaneous firing of action potential.
Okay. All right, one last question for Laszlo. The paper on HCQ is very interesting. This is a compound that was developed by Novartis many years ago, correct? Can you comment on that? And that there were no safety issues, but that was with HCQ alone, correct? And which therapeutic area?
Yeah, okay. So HCQ, hydroxychloroquine, was developed many years ago, and it became a staple for preventing of malaria. So we know it has, and we knew it has effects at various ion channels, as I showed you. But the safety margins in healthy people, healthy volunteers, is considerable. It's over 30 for all these ion channels. And because it was basically used as the preventative treatment for people who travel to areas where there is a malaria endemic, it was applied to that, and it is still applied. So it certainly was a repositioning effort, and don't quote me on this one. I don't think it was Novartis who did it. It came from the clinical studies that somebody tried it, and they believed that it was working. And I think there was some study from Chile or Peru, which seemed to be confirmative that it would work in COVID-19 patients. There's a bewildering amount of information in the publications about it, but the original development was for prevention of malaria.
Okay, thank you very much. Well, Laszlo and Najah, thank you for providing an excellent webinar. I'd like to thank Gary Watkins, Director of Marketing at AnaBios, for putting this together, and everyone in the audience for joining. Again, this has been recorded and will be available in a few days. Stay tuned for check regularly for the next webinar, and we'll see you then. Thanks, everyone.
Thank you.
Thank you very much. Take care. Bye.